Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of the show. Today, bringing you another really awesome guest involved in creating a better tomorrow for millions of people out there. Today, we have the honor of being joined on the show by Dr. Laura Aguilar, who is the Chief Medical Officer at Diakonos Oncology. This is a clinical stage immuno-oncology company. They're dedicated to revolutionizing cancer immunotherapy, focusing on these really difficult to treat indications like glioblastoma. Dr. Aguilar is a physician scientist and a biotech entrepreneur. She has over 2 decades of experience in clinical and regulatory development of these really complex biologics for cancer, with a focus on solid tumors, including glioblastoma and pancreatic cancer experiences. Dr. Aguilar previously was co-founder of Advantagene, which is now Candel Therapeutics, and we profiled their oncolytic viral program a couple months ago, and there she served as chief medical officer and was also the leader in their strategic development from inception to IPO, oversaw broad clinical development of that company on those viral immuno-oncology products. Dr. Aguilar is also attending physician at Dana-Farber Cancer Institute and Boston Children's Hospital, and associate director of the Harvard Gene Therapy Initiative at Harvard Medical School. She also currently serves on several boards, including the board of the American Brain Tumor Association. She did both her medical degree and Ph.D. in microbiology and immunology at Baylor College of Medicine, and completed her clinical residency in pediatrics and fellowship in pediatric hematology oncology at Texas Children's Hospital. A lot of very important topics to get into today. Really honored to have her with us. Dr. Laura Aguilar, thank you so much for taking the time to come on the show.
Thank you for having me.
It's great having you. Obviously, a set of very important topics, and I'm really looking forward to the discussion today. Would very much like to start off, at the beginning, is reading sort of all about you and your sort of early work in the area of immunology and on everything from T cell receptors in the adult thymus, these thymic nerve cells. Really interesting set of topics. But it's not until this clinical trial in I think it was 1998, where you sort of add the oncology into the mix, and this was you were working on these Epstein-Barr specific cytotoxic T lymphocytes. Would love to hear about the early story here, because you have clearly been involved in many interesting complex biologics for cancer, but tell us a little bit how it all started off, if you would.
Well, so I originally thought I wanted to be a veterinarian, but during undergrad, I did a research in a cancer biology lab, and it was in the early days of trying to understand cancer and viruses' role in cancer, and so that kind of got me into the kind of thinking about that link. And then I ended up changing my mind and deciding to go to medical school. And I did an MD PhD at Baylor College of Medicine, and had a great immunology professor in my first year of medical school. And so I ended up deciding to do immunology as my PhD. And I chose a lab that was actually in the genetics department. And that really helped me get involved in kind of an early wave of applying molecular biology to immunology, when most of immunology before that had been cell-based. But there was also a strong cell-based program there. So I kind of got a little bit of both, but got really in on the cutting edge of that technology wave. And it was also a big gene therapy place at Baylor College of Medicine. The whole thing was just starting. And so I started being, right in the midst of all of that in the early days. And then I finished my PhD on the thymic nerve cells, as you described, and finished medical school. And then when I went to do and did my residency in pediatrics there, because that I was really drawn to working with kids. And then when I did my fellowship in pediatric hem-onc, there by that time, gene therapy was starting to be in the clinics, and
Yep.
early immunotherapy was starting to be in the clinics using viruses to try to stimulate immune response against tumors. And so that was all very exciting. And my husband, who I'd met in the lab actually back in undergrad, he was running one of these labs where they could make viruses that you could use to treat cancer, and starting to study that in clinical trials. So, I was kind of right there in the midst of all of this as it was starting. And I started I did my fellowship research in the lab of Cleo Rooney there at Texas Children's, where they were working on taking cells from patients, in that case B cells, modifying, using those B cells to stimulate T cells in
Hmm.
culture. And then we gave the T cells back to the patients. And those were cells that were targeted to recognize an antigen from the EBV virus that is on some tumor types. So it's not dissimilar from what Diakonos is doing now.
Right.
But this was, in the days when CAR T approach,
Yep.
where you use T cells, but you have to modify them to make the T cell receptor recognize a specific antigen. We were at the time doing this with the viral antigen.
Hmm.
So that kind of got me into the role of translating things from the lab to the clinic, doing everything from taking cells out of the patient, growing them in the lab in a specialized GMP lab where you can do that in a way that you can give it back to a patient, and
Hmm.
then actually being the one there giving it back to the patient and then doing all the analyses to see how it was impacting, how long were those cells lasting, was it affecting their tumor and all of that.
Hmm.
So that kind of started that journey, and then I we moved to Harvard and started doing something similar there and trying to get some trials going there. And unfortunately, it was at a time when there had been some failures in gene therapy and some unfortunate safety issues, and so it was really hard to get things going in the clinic. Funding was really hard to get. And but we, made some good progress there while I was also seeing patients. And what I found when I would see patients in the clinic, which I no longer do, is that, the patients that I would see with a cancer that I knew that we didn't have a good treatment for, we would give them cytotoxic chemotherapy, we would give them, radiation and other things, and the patients we knew weren't going to be cured by this. And especially in kids, it's just a really hard thing to accept.
Hmm.
And it felt like there should be something better. And this approach of using a patient's own immune system to fight against their cancer was something that just made a lot more sense. And so that was in the days when immunotherapy was just in the early days of being tested. The checkpoint inhibitor approaches hadn't yet come on to be shown to be effective. And so it was this exciting time and also a challenging time because it
Right.
was very difficult to get support from funding agencies. And so we ended up, that's how I got involved in the co-founding of the company, actually with my husband, started in our basement. And this was using a virus to stimulate an immune response against cancer. And then, came full circle many years later to Diakonos, where we're taking cells out of the patient, kind of going back to those early days at Baylor. Curiously enough, the technology is actually being developed at Baylor College of
Yep.
Medicine, just a coincidence,
Hmm.
to try to do the same type of thing, stimulate an immune response against a patient's cancer, but do it with their own immune cells.
Hmm. Hmm. Yeah, it's again, it's a lot of the parallels looking back to that early work. Um, Before we get to Diakonos, I just wanted to ask you about one other thing because obviously you mentioned you're working with your husband on some of these gene therapy products, and I saw some of these papers from sort of the early 2000s, where you talk about sort of the challenges when it comes to neuro-oncology, and I know you did the some clinical work with this the interesting the herpes simplex virus
Mhm,
with the ganciclovir kill
mhm,
switch or whatever, I don't know the right terminology here.
mhm,
But in when you were doing that work, you also wrote several papers about, hey, this is challenging stuff, not just immune response related, getting this complex biologic right, but also getting things into the central nervous system. And just love to hear just a couple, top line learnings from that period, because we're going to be getting into gliomas and glioblastoma and so forth, still
mhm.
really tough cancers. What were some of the early learnings in terms of the neuro-oncology theme that you ran into then?
I think that, there's been this concept of blood-brain barrier, which is that in the space of the brain, that the brain is protected from the immune system in a way and from other toxins getting into the brain. And so this has been a topic, it's still a much debated topic about when a patient has a brain tumor, is that blood-brain barrier broken down such that things can get there or not? And so in the early days, we didn't really know if T cells could get there when try to do an immunotherapy. The viral approaches have had the advantage that they're being delivered directly to the tumor, and there's many different groups that are doing this. So you knew you were getting it to the tumor because that's where you were putting it. Whether the T cells could get there or not was something we didn't necessarily know, but we found from being able to look at patients' tumors after treatment, we could see that the T cells were in the tumor. So, and they weren't there before the treatment. So we knew that that could happen. I think, you know, one question that I think we've gotten more clarity on is why do some tumors respond to immunotherapy and others don't?
Mm-hmm.
Glioblastoma being one that, at least with the checkpoint inhibitor immunotherapies that are approved for many different cancer types now and are very effective, have not been effective in glioblastoma. And what has become apparent is that glioblastomas are very immune suppressive. So you probably need more than that one signal. A checkpoint inhibitor basically takes the brakes off
Yep.
of, because when you stimulate an immune response, there's a reaction to keep it from becoming a bad immune response that attacks yourself, an autoimmune disease. So the brakes are there for a reason in biology, but if you take the brakes off, but the car was just sitting flat and didn't or have any, momentum,
Mm-hmm.
it's not going to work. So that's one part of it, is that glioblastoma is so immunosuppressive that it really needs a strong immune stimulatory signal and possibly also something to take the brakes off. But that is not really clear yet whether the combination would work. So I think those are kind of some of the things. Unfortunately, the standard of care in glioblastoma hasn't really changed in
Yep.
many years. It's still surgery followed by radiation and chemotherapy with temozolomide. There have been many trials trying many different things, and unfortunately, it really hasn't changed. And one of the things that I think we also kind of learned early on or an approach was that we would like to try to get at cancers like this, challenging cancers like glioblastoma, pancreatic cancer being another one, early, so that while patients still have more of a healthy immune system, there's more of an opportunity that they might be able to fight off this cancer if we give them the right tools for that. Um, and that is kind of something that is harder to do when you're trying to test a treatment if you're doing it in combination with the standard of care early, because you have to wait to see, and you really need a control group to compare whether it worked or not. So those are some of the challenges that we've spent a lot of time on previously, and I continue to do at Diakonos, is how can we develop novel trial designs and novel endpoints that allow us to get to a meaningful understanding of whether the treatment is actually benefiting the patient while you're giving it earlier and in combination with standard of care.
Right. Right. Excellent. Perfect. So laying that groundwork, let's get into the area of dendritic cells. And, the last time we talked about dendritic cells on the show was with Drew Weissman around the corner here at Penn, specifically focused on dendritic cells as a presentation mechanism for vaccine development. A very unique type of cell compared to, say, the CAR T work, in the sense that here we have a cell that sort of it bridges the innate and the adaptive immune systems. It is very good at presenting antigens of both foreign type, so the microbes and viruses and all that, and weird antigens that pop up on cancer cells and other nasty cells in our bodies that we don't like. Take us into the world of the dendritic cell, sort of reintroduce us to this theme, a little bit of what they normally do in our bodies. And again, this principle of which, and we'll get into sort of how you double load, but this concept that it can present two different types of antigens in doing its work. But when it does present two types of antigens, it sort of becomes a really powerful dendritic cell and does so much more for us. So I'll let you run with that one and take your
You're
time.
making my work easy for me now. You've done your homework. Well, so dendritic cells, if you think about kind of how the immune system is regulated, dendritic cells kind of sit on the top of the hierarchy, and they're kind of like the master regulators. And they help our immune system kind of know what it should respond to and what it shouldn't. So they regulate the downstream effects, which is a lot of it, you mentioned innate immune system, as well as T cells, which is kind of where we focus in cancer a lot, B cells that make antibodies that circulate in the body that can attack things, and then the other cytokines and other pieces of the immune system that help us fight against foreign invaders. But also it regulates so that we don't end up with autoimmune diseases. And our body, our immune system is constantly fighting against mutations. So cancer cells pop up all the time in all of us when there's a mistake that happens in the DNA when cells replicate. And most of the time that gets cleared by our immune system. Infections come and we respond to them and we clear them, and then the immune system quiets down like it should. We don't want to continue to be in a hyperactive immune state like we are when we have a bad cold forever, right? We want that inflammation, everything to calm down. So that's something that the dendritic cells help kind of regulate. And so it's a place that makes sense to start if you really want to orchestrate a good immune response against cancer, because that's kind of what's controlling everything. And the dendritic cells, as the way that they fight viruses, is that they are able to present antigens from the viruses in different ways. So you were mentioning the two different ways. So on the dendritic cell, there's MHC molecules that are two types, class 1 and class 2, and these present antigens kind of from different sources. So class 1 presents antigens that are coming from inside the cell. So like a virus that's inside of the cell will make antigens from its own. The virus could be a DNA virus or an RNA virus, and those turn into and make antigens inside the cell. And then they also have this MHC class 2, which can take antigens from outside the cell that are proteins that are floating around. The virus may be floating around and has proteins on the outside of it, and it can take them in that way. And so, what is kind of an interesting discovery that you were alluding to is that what Diakonos found is that if you were to load dendritic cells with antigens coming from both pathways, that this really stimulates a much better immune response. And other people have done similar things with cross priming and how do antigens get there, but I've not known of any other group who's done this where they load them the way Diakonos does. It's similar to some of the viral immunotherapy approaches where in that case all these tools are being put right into the tumor, and dendritic cells can come in and present antigens and do all of that. So
very cool. Yeah, and as you were saying, this is the initial word came out, Texas Medical Center, that the company is based on. And again, we'll talk a little about this principle of double loading. And again, I'm just taking this some of this information off the website. What we're dealing here in terms of the Diakonos process is a personalized, individualized method by which someone's tumor tissue, the antigens are collected, and I'll let you walk us through this. The double loading effect then happens in the lab with these dendritic cells. There's some magic to make a lot of them and then brought back to the patient. Talk a little bit more about that if you would, and specifically, and if it's proprietary, you don't have to go into it, but in terms of the infectious part of the double loading, can you talk about what's used there? Is it some virus or unknown a virus? Is it some lysate of stuff? What is typically used for that as opposed to the neoantigen part of the loading?
So, so there's different ways to present antigens to T cells, and different ways to get them onto those antigen presenting cells. That we're using dendritic cells is what we're talking about here.
Mm-hmm.
So when you put when you're vaccinated with flu or COVID vaccine that's given intradermally, that is actually getting taken up by things
Yep.
like dendritic cells that are in the skin. And so that's kind of doing the same idea. In the case of, when we're dealing with a difficult cancer, like glioblastoma, where there's so much immunosuppression that we need to overcome, we need to kind of maneuver the system to give the immune system a better chance. And so the way in which the discovery that Diakonos is based on is that it takes a patient's own tumor, so you're getting all the antigens, but like in a glioblastoma or pancreatic cancer where we know they're very heterogeneous, so there's not just like one antigen that we can target, or
Right.
some other groups are using a set of antigens.
Yep.
Moderna is doing work with an mRNA vaccine where they figure out a set of antigens from a patient and use those.
Right.
And what we're doing is taking all of the antigens from a patient. Just they take a piece of their tumor and use that as the source of all of the antigens. And in order to load this onto the class 1 and class 2 and mimic what it would happen if it was a viral infection, we're taking the mRNA from the patient's tumor, so it's their own mRNA in their tumor, and amplifying it so that we have enough to be able to load dendritic cells with it. And then we're also taking the tumor tissue and gets processed so that it makes peptides, basically. So
Mm-hmm.
that's the loading onto the class 2 part. And so it's really all coming from the patient's tumor being processed in the specialized GMP lab to make those two pieces, but it's still all coming from that patient's own tumor. And then the patients go through a process called leukapheresis, where we collect their monocytes, which is really the precursors of a dendritic cell. And
Sure.
those then go to that same GMP lab where they get processed so that it kind of selects out the cells that we want in the culture process and stimulates them with some cytokines to get them revved up and
Right.
and matured. And then they get loaded with that lysate, the peptide part, and the mRNA from their own
Okay.
tumor. And then that gets frozen into, little individual vaccine vials for that patient. So it's a personalized vaccine for the patient. And then that gets sent back to the hospital or clinic wherever they were originally, were on the clinical trial where they had their tumor surgery and they had their leukapheresis done. And then it gets delivered near lymph nodes. So lymph nodes are kind of where the interaction between antigen presenting cells and T cells happen, as you know. And so the the idea is to get this as close as possible to where the tumor is so that because the draining lymph nodes, the regional lymph nodes, we have lymph nodes all over our body, the ones that are closest to the tumor is kind of where the action's happening and where
Hmm.
we want to stimulate T cells so that they can now go to the tumor and attack the tumor.
Excellent. Excellent. And you did mention one of the things I missed there was the additional sort of side, and I think you use interferon to
Yes.
even amp up more this response. So
Even
no,
amp up more, exactly. I mean,
that's
that's what
that.
these kinds of tumors, it's really necessary to do as many, things that so we can to overcome that immunosuppression that's
Right.
happened. And so the interferon, that's actually an interferon that's used to treat hepatitis viruses um, in combination with antivirals, and it helps to stimulate the immune response against the cancer, and it helps the T cells that we've stimulated continue to be more active. So they don't get kind of shut down as quickly as they might if because of that counterbalance I was talking
Uh,
about. This kind of helps keep them revved up for longer.
Yeah. Yeah. No, that's again, we're learning so much about the immune system and all these different players that, again, the fact that you're, focusing on each part of this issue is extremely important. And the other, just to reiterate for the audience, this principle that you're losing all the antigens, because we think so much about sort of targeted therapy as, oh, got this one weird gene out of whack, but no, we know more about the heterogeneity of these tumors now that we want the immune system looking at lots of potential antigens on these tumors as they grow and change and do their things. So this
Right.
is very
And we
important.
know from some experiment, some trials that have been done that human tumors are very good at evolving
Yeah,
around a treatment. So a
yeah,
targeted treatment, even a tyrosine kinase inhibitor that targets a specific mutation, they patients tend to become resistant with time
right.
because the tumor evolves. And so that's particularly a problem with targeted immunotherapies against a single antigen is that they can become resistant, and the cells that no longer express that antigen, that mutation could keep growing. And so the therapy wasn't really effective against the whole tumor. So by taking the whole tumor and stimulate that immune response to all of it from the beginning, we think that we can keep the patients in a responsive state with their T cells, keeping their tumor in check or hopefully curing it.
Mm-hmm. Excellent. Um, and as mentioned at the beginning of the show, Diakonos, its mission is the difficult to treat cancers, and you have chosen the probably the worst of them all, glioblastoma, as an initial target here. Just a couple of months ago, you presented phase 1 data, and again, this um, you could take us a little bit into sort of the 12-month survival and all that of these patients, but it's horrific numbers normally with standard of care even. Walk us through the program that is known as DOC 1021, a autologous dendritic cell vaccine for glioblastoma, and some of what you saw in this phase 1 study, because it's pretty remarkable, and I'm excited for what's happening next.
Yeah, I think it's something about small companies. They just want to do the hardest things. I don't know why it is. Sometimes you don't know any better, so that's where it starts. I think
Right.
that's how it happens sometimes. Naivete maybe. You find colleagues that want to do something. I think sometimes when you're starting something new and novel,
Yeah.
it's just sometimes easier to get going in something that is such an unmet need and unfortunately so devastating. And it helps drive you through those hard times when it's hard doing these trials, and it's hard, especially getting a company going when it's hard to raise money and they're small and everybody has to do everything. It's a challenge, and it helps to be driven by the passion that there are patients out there that really don't have good treatments, and that's what keeps us inspired, keeps us going. Um, and yeah, I just saw this morning about Mia Love, who was a former congresswoman from Utah,
Yep.
just passed away from glioblastoma at 49. So, another, I mean, it's not a common disease, but it seems like we keep finding, knowing people that have had this disease. So that really is our inspiration. And so Diakonos this did a phase 1 trial. So phase 1 being the first in human study where you test whether the safety of something. They also had started a study with pancreatic cancer early on as well. That's still ongoing, but another hard one. But glioblastoma started a few years ago, and at that trial, and has finished enrollment. And this is what kind of got me involved last year in this with this company. So the phase 1 trial is for patients with glioblastoma and adding to standard of care. So they're still getting their surgery that they would normally get. And like I said, that's where we get the tumor sample to be able to make the vaccine. And then they go ahead and get their chemoradiation that's standard of care after the vaccine, I mean, after the surgery. And then after they finish 6 weeks of standard chemoradiation, they get their vaccine, 3 vaccinations 2 weeks apart with that interferon you mentioned. And then they go on to get their standard of care chemotherapy that's given for about 6 months after that. And what's really been impressive in this phase 1 study is a few things. One, it was really safe. So patients, we know that they have some symptoms that they're having an immune response, which I think is a good thing. So, mild flu-like symptoms, chills, a little bit of like nausea, malaise for a short period of time. Um, and a little bit of swelling in that area where they got the injection near their lymph nodes. So not surprising, we're stimulating immune
Sure.
response in the lymph node. You're going to feel a little bit of that. But again, that was short-lived and not no serious events attributed to the treatment.
Hmm.
And what we're finding is that the patients seem to be there. We have a group of patients that, some patients are living longer than expected. So we have patients that are lived past 18 months, even 24 months, more than 30 months. This is just unexpected. And it also, depending on some of the characteristics of the patients, we can see that these are not patients where you would expect that given that patient's
Right.
characteristics. So this has been, exciting. We've also been able to see that some of the immune stimulation that we were expecting is happening. So some patients that went back to the operating room were taken back to the operating room, and when it looked like their tumor might have been coming back, what we've found is that there were T cells in their tumor. So that's a sign that we were getting that immune
Sure.
stimulation from the dendritic cell vaccine, and the T cells were able to get there and get into the tumor and do their work. And we also know when looking at a patient's blood after treatment, we can see activated T cells in their blood that weren't there before they got the treatment. So some signs that it is doing what we expect it to do. There's still a lot more that we'd like to interrogate about that to understand better. But overall, it's been encouraging enough to get designation from the FDA for a fast track designation, an orphan drug designation. These are designations that help us speed up the process to get a new approach through the regulatory steps that are required, and helps us have more conversations with FDA about that so we can be more creative and thoughtful about how we do this. And that's given to things that have a serious disease with an unmet need, where there's not a treatment that is adequate to prolong survival,
Hmm.
and that um, that is showing some signs that it could work. So that
Right.
is very, it's validation that the FDA was seeing this as promising as well.
Hmm.
Um, and so that was the phase 1 study. And now what we need to do is the next stage. And there's phase 2 and then phase 3, which are the next steps. Phase 2 is to determine if there's efficacy of the treatment in a bigger trial so that, that's more controlled, and it may be one dose level or a couple dose levels. And then phase 3 would be to do a definitive trial to prove that it works. That often is a placebo-controlled randomized trial, because before the FDA will approve a treatment to be on the market um, for a given indication, that needs to be really proven with very strong statistical evidence.
Sure.
Um, we're we've designed a phase 2 and it just opened last. Several patients already signed up last week to test this, to do this phase 2 trial, and we've designed it in a novel way that might even be possible if it really looks good. That could even kind of jumps through the phase 2, so you can do trials where you kind of combine the phase 2 and phase 3 in an indication like this that's so devastating and
Right.
where there's such an unmet need.
Yeah. It's interesting. I had a friend of the family that recently lost a battle with glioblastoma, and they were, and not to say that there's worse. I mean, it's all bad, but he had, the advanced stage. There was the meninges, the cerebral spinal fluid, and just horrible. And it got me thinking because I was looking at some of the materials per the phase 1 study, and you were commended as a company for including a lot of patients in the study that were, I guess there's a lot of exclusion criteria in the past that may have been used, but talking about advanced patients, talking about older patients, which might have back in the day. Okay, we have a cutoff here, but no, nowadays things are different. I just love your perspective as a clinician who's been in this space for a couple of decades now. What are some of the things you're seeing in terms of inclusion criteria, how things have changed in this era of these novel types of
Mhm.
products?
Well, I think you're right. Age is one that age is, there's not a strict criterion where you can say anybody above this age doesn't have a good immune system or
Right.
isn't going to tolerate. It depends on so many other factors. So we don't have an upper age limit for that reason. We can't just blanket know where to cut that off. We include patients we'd like for patients to have a really good resection of their tumor as much as possible, but sometimes depending on the location of the tumor and the neurologic effects it would be that they would have, side effects from certain parts of the brain, if they were resected, would cause really difficult side effects. So sometimes it just can't be as much of a resection as you would like. And some trials will exclude those patients. We're allowing them down to a reasonable level. We still
Mm-hmm.
have to pick a setting where we think we could have a chance of helping the patient, because it doesn't make sense to give the patient something that that just we don't think will work. So
Sure.
we have some selection, but not too much. Not too many exclusions. We've something I think that has evolved is how to deal with HIV patients. So if a
Mm.
patient has HIV, but they're fully on effective treatment and their T cell levels are good and their virus level is undetectable, they should still be able to be in an immunotherapy trial. That's
Right.
not something that has always been the case. People in the past typically excluded any patients with known HIV. So I think there's been good studies to show that is not necessary.
Got it.
Um, and we're also trying to have a pretty diverse population of patients in the trial, and that the easiest way to do that is to have sites for the trial that are in all kinds of different parts of the country, so that it's not just academic centers in places like Boston and San Francisco, which we are including, but we're also have sites in different parts of the country where we might have a catchment that is a little different. We're looking at sites in Texas and multiple places in Texas and Alabama, and kind of a community hospital setting in New Jersey, but in not just like the major academic medical center, but one that has a catchment for a wider group of patients that often aren't included in clinical trials.
Excellent. Um, Now, it's not like for indication two, you took anything any easier, because pancreatic adenocarcinoma is your second indication, which you recently received fast track status from the FDA, and I think a lot is based on the great stuff they saw with glioblastoma. But again, in those programs, it's a little bit earlier. But if you could say a few words about what's happening on this front, I think that'd be really interesting as well.
Haven't picked the easy ones.
No, you didn't.
And I think that's again a sign of a young company that is a little altruistic maybe like
I love it.
but that's how advances get made. So
Absolutely.
pancreatic cancer it's analogous, I guess. I mean, I worked on pancreatic cancer as well as glioblastoma for many years, so kind of have that same experience of getting into the hard ones. And it is also there are some analogies that it is a tumor type that has not been shown to be responsive to immunotherapy.
Mm,
And we kind of know, like there's data on what tumor types tend to respond to immunotherapy and what don't. There's like a spectrum. And one of the things that's known about why that happens, besides immunosuppressive nature of the tumor, is also mutational burden. So cancers like lung cancer, melanoma, some other cancers that have like environmental or mutagenic uh,
sure.
causes, they tend to have more of a mutation, more mutational burden, and those antigens are very actively stimulate the immune response. Pancreatic cancer, like glioblastoma, is one that's on that further on the low end of the mutational
Hmm.
burden spectrum, which makes it typically not as responsive, immunotherapy responsive. We're doing this similar approach to glioblastoma where we're trying to go up early.
Hmm.
So patients who have metastatic pancreatic cancer and maybe have already had several treatments that haven't worked, that's a very difficult place to expect an immunotherapy to work. And unfortunately, there have been many trials that have been done and that haven't worked. So we're going back to the early, newly diagnosed patient population and patients who are getting a surgery. So their tumor is still enough contained at that time that they can have a surgery to remove it. That gives us that tumor sample we need to make the vaccine. And it also means that when we're vaccinating the patient, they don't have a huge amount of tumor all over their body. It is in what we call a minimal residual disease state, where
Mm,
it's a good time to stimulate an immune response. And then they still get their standard chemotherapy because we need to give them all the chances that we can. And what we're hoping is that by stimulating that immune response, we know in pancreatic cancer, it always ends up metastatic, really. I mean, almost always. So there are tumor cells, even though we think it's all in the primary site and it's all resected, it comes back because there were some cells somewhere that didn't get removed. And it's not, chemotherapy is not that great at clearing all of it.
right.
So if we could stimulate an immune response, we have T cells that can go around and kind of mop up what's left
Hmm.
and keep it kind of in check so that it doesn't take off and regrow and start making metastases somewhere else. That's where we think we could really most likely help these patients.
Um, I just want to ask a question related because clearly these therapies, whether we're talking about gene therapy or oncolytic viruses or what you're doing now with dendritic cells, these are not the therapies of yesteryear, right? I mean, they're living and they're very complex and heterogenic and so forth. Um, any interesting stories, insights related to, I guess, I don't know, it falls into CMC, uh, the traditional concept of CMC in developing a drug, but along the way, have you found that regulators have gotten it's become easier to bring non-traditional therapeutics of this nature to the agency from that sort of the production perspective. Obviously,
Mhm.
they want to see the data, but at the end of the day, my the few times I ever spent was down at FDA. It was like, hey, how do you manufacture this thing really well, standardized constantly? And again, you're dealing with things that are not traditional and like to
Mhm.
change, let's say, when they're being cultured and so forth. Any interesting insights from sort of the
Right.
CMC angle?
I think that it, I mean, my experience, and I'm not the CMC person, but
Understood. Understood.
I've been near it a lot.
Yeah.
Both my husband, who was a leader and has been and still is a leader in that field of
Sure.
how to make viruses in a way that you can use them to treat a patient from the days when FDA didn't know how to regulate that because it hadn't been done. Similarly with cell therapy, how do you regulate something like this that doesn't, it's not a small molecule that you can just
Right.
purify, sterilize and do all that. So but the same principles are applied and they're applied in the process. And they have to be applied with logic and quality control so that you're consistently doing the same thing every time as much as possible, which is sometimes hard with a living cell that comes from different patients. It's not
Right.
always perfect. But you control as much as you can and you test for things. And you test for things at different steps because a virus infection in the cells in culture may not be that easy to detect at the very end, but you might be able to pick it up earlier at a stage when that's more likely. So just being smart about those things. And, I guess it, the agency, the FDA has, in my experience, has been very good about working with sponsors um, who are trying to do these cutting edge things, being open to hearing they really expect you to propose what you think makes sense. They don't
Hmm.
tell you what to do.
Right.
You propose, and they can tell you yes, no, look at it slightly differently. They'll send you to the regulations, but the regulations often say you should do this, you should do that. You know, the quality needs to be there, but it's not always too specific about exactly what you need to do because those regulations have to apply to so many different things.
Right. Right.
So I think having active conversations, and that's where things like the fast track and orphan drug designations really help because that's their mechanism to have a lot of those
Hmm.
frequent interactions. And and they're smart people at the agency, um, and so but they're also busy and they see many things. So being able to help them by providing the information to them in a easily digestible way and a thoughtful way that you've already thought through as much as you can so that they can give you good feedback on it. And also looking at risk benefit ratio because what you could do in a first in human phase 1 study is different than what you need to be doing by the time you're in phase 3 and trying to
Sure.
commercialize. And
Sure.
so you don't need to solve all of that when you're in phase 1, but you need to start thinking about it so that you would be ready once you get there.
Absolutely. Absolutely. Again, I appreciate that insight on that front because, again, thinking back a few careers ago, that yeah, bringing these things that are not traditional, small little white pills. um, but no, I'm really glad that there's that flexibility now.
And the other piece that I'm always harping on is how do you get it actually to the patient? Because
Right.
you have something, you know, sitting in a freezer and maybe it was tested and you knew that when it's at that, -80 or whatever that temperature, liquid nitrogen where it's being stored, it's stable there.
Right.
You take it out, how long can it be thawed? What do you like? How quickly does it need to be diluted? What can it be diluted in, and how long can it stay in that before you get it into the patient?
Hmm.
Those are such critical factors that are sometimes missed, and it you can end up delivering something that's not even effective if you're not control testing those things and controlling those steps. But it's all
Hmm.
doable, right? I mean,
Right.
when CAR T first came out as a therapeutic, I thought, oh my gosh, how are they going to do this? Like as a drug, as a licensed product, that sounds really challenging having grown T cells in the lab myself. And technology can be advanced. You can find better ways to do the process development, more efficient ways. Things can get automated, with time
Hmm.
those efficiencies can be built in with creativity and enough funding.
Yep. Yeah. That's always the case, isn't it? No, but no, but clearly, you're being very creative and coming up with a really novel solutions to these horrible diseases. It's just it's very impressive. Again, not just what you're doing now, but your entire career and sort of thinking about these problems. Laura, what else should we be on the lookout in terms of Diakonos for 2025? Clearly, you have the glioblastoma program, the pancreatic cancer program. I saw a program in sarcoma as well. Any other public facing stuff that we could know about while we have you today, or anything I missed while we have you
So
today?
yeah, well, since I joined, I looking at the portfolio of glioblastoma, pancreatic cancer, we've being, as we've discussed, difficult cancers to treat. And that spectrum I was telling you about, from higher mutational burden being immunotherapy responsive, to the other end of the spectrum where glioblastoma and pancreatic cancer have not. We are planning a trial in refractory melanoma. And so
Ah,
this is in the space where it is known to be immune responsive. This is where the checkpoint inhibitors got their initial approvals. In this case, though, instead of going early, where the checkpoint inhibitors work really well, that's the standard of care, we're going to the refractory stage. So after patients have gotten their
mm,
standard of care treatment, but some patients are cured, and some patients, their cancer is refractory to that treatment. It either responded and then stops responding and they start having more tumor growth and maybe more metastases, or they just never responded to a checkpoint inhibitor. So they're shown to be resistant to that. And so this is a place where we know there's lots of antigens there. We know that the tumor itself can respond to immunotherapy. And the checkpoint inhibitors in those refractory patients aren't enough. And
mm,
so adding the dendritic cell, the DOC 1021, to that setting in patients who are not responding to their checkpoint inhibitors, see if we can turn them into responders, like get that car, moving, tip it down the hill so that it's moving, so that their immune system can get back to fighting their melanoma. So that is something that we're working on. It's a space where other groups have tested things. That there's the TIL therapy, the tumor infiltrating lymphocyte therapy, came out.
mm,
That's from the NCI, that was developed at NCI. That came out a year or two ago. I got approval, I guess it was last year. And so we know it can be done. We kind of know what to expect. And the other advantage in that is it's a little easier to see what's happening. So we want to be able to really interrogate how are we how are we impacting the immune response?
mm,
Because in a melanoma, it's more accessible. So, and they're typically metastatic by the time they're in this state, and there may be multiple sites of disease. And so we can give the vaccine and then get a biopsy from different areas afterwards. That's a hard thing to do in glioblastoma or pancreatic cancer. We can get biopsies and also get blood samples and see what's happening. Are all the patients having an immune stimulation, or just some patients? Are the ones that their tumor shrinking, are they the ones that are having more T cells in their tumor? How can we kind of figure out who's responding, who's not, and why, and understand the biology better?
mm.
So that's a place where we think we can really like learn more and hopefully help patients as well by seeing their tumor responding in a way that you can visually see it, or it's easy to see on imaging. And that's something that's difficult in glioblastoma and pancreatic cancer.
Sure, sure. And it's another uh, significantly unmet need. So
It is, it
again,
is.
and just another impressive aspect of the portfolio. I just yeah, commend you for this. Um, but
It's really a platform technology. Any tumor that we can take out and make a vaccine from could be treated. in order to really test efficacy for something, we need to um, define the patient population to some extent, be as open as we can, but we need to be able to see any actions with other treatment that those patients are getting and characteristics that we know about that tumor type as far as what is expected so that we know what did our drug do when it was added to all of that
Yeah.
other, they're not mice. We're not
Right.
mice. So
Right.
we're not inbred mice that have a set genetics and control where we can control the environment and all of that. So we have to build that into how we design the clinical trials to control as many variables as we can.
I tell the story of my father-in-law occasionally on the show because he was on one of these radionucleotide antibody drug conjugates a couple of years ago. We had to stay away from him for, for a few days after each of his administrations because he make the house radioactive. But yeah, it's
Mhm.
yeah, again, a lot to think about. But clearly, throughout all these experiences that you've had and these different types of products that you've been working on, you know you've gathered such important knowledge, and I commend you due to the portfolio. I really am excited about what happens next, and just looking forward to the success of the company in these and other indications that it gets into. Laura, I just wanted to, while we have you today, I just wanted to loop back now just for have a little fun. And at the beginning of the show, you mentioned that before you thought about immunology and microbiology and all this stuff that you wanted to be a veterinarian. I was listening to a presentation that you gave. This was, I think it was last year at some point. You were talking about some of these things, but you were also talking about, I think, your daughter, who is not just is working with lions and other big creatures. And I was just, it's just an interesting little side story. But does she take you on any of these trips? Do you get to hang out with lions at all, or
Yeah,
what's it all?
so it's funny how things happen.
Yeah.
So I have two. One is a medical student at Harvard, and
Uh-huh.
so I think got the bug from all of the things that we did maybe, but I kind of found their own path to medicine and is going to do great
Cool.
things for patients. Um, and then um, and then the other one is a graduate student at Harvard and in a PhD program looking at studying it's like evolutionary biology and
Uh,
um, organismic and evolutionary biology is what they call it. But
yeah.
she's studying the lion-human interactions in Kenya. So
Wow.
trying to get at kind of the science behind why does conflict happen that leads to lions being in such so few lions and as an example of species that are really threatened. And so she puts collars on the lions to be able to track them, and she's also talking to the local people that herd their cattle and putting tags on the ears of cattle so she can follow where they are, and then doing LiDAR mapping of the area, and then she can superimpose where the lions are at various times, where the
Wow.
cows are, where the people are, and what the vegetation looks like, and try to understand also culturally, doing surveys of the local people to understand their perceptions about lions and how
Wow.
to avoid lions being killed if we can keep them from being such a
Sure.
threat to people. So
Sure.
yes, and we did get to go in February to visit and we're able to attend a few exciting lion collarings. So
Nice. Nice. Well, that's pretty cool in itself, but I can imagine your sort of dinner conversations with all these amazing minds around the table. But no, it's just another really cool part of your story, and I just wanted to know. I thought I'd bring it up there. But no, really amazing stuff. Again, I look forward to continue to follow the success of the company and wishing you and the team the best with all these indications. Again, for everybody that's going to be listening to this episode of our show across the various podcast networks or who'll be watching on the YouTube channel. Again, you've been spending time with Dr. Laura Aguilar, Chief Medical Officer, Diakonos Oncology, doing really amazing things to revolutionize cancer immunotherapies, treat these difficult to treat indications like glioblastoma and pancreatic cancer and melanoma. Laura, again, I want to thank you for taking the time out of your schedule to talk to us for a little while today and educate us on all these topics. Thank you for everything you're doing for patients. And as we like to say on our show, thanks for helping to create a better tomorrow for so many people out there via the type of work you're doing, and continue to do really an awesome story. And I look forward to continuing to follow it.
Well, thank you very much. I appreciate the opportunity to spread the word about this.
Really great having you.